Okay, we'll get started. So welcome to this class. So in this class, we'll just be essentially cramming as much of Biology Paper 1 just prior to the exam tomorrow. I've got a few exam questions. There's a few certain topics that we're going to cover as well that kind of got a rough idea probably will come up just because they've not come up for years. So we'll jump straight into it. And if you've got any questions as well, the chat's open, so you can drop any questions in the chat. Everything that will get written today as well, I'll send it out straight after, and this is getting recorded as well. So if you do want to look back at this, you can do that as well. But yeah, ask any questions as we're going as well, and hopefully you'll gain quite a lot out of this. So let's get started. Yeah, this is so what I'd say for Edexcel, just make sure you compare it against your content list, just to make sure its topics are in tomorrow's exam and not for after the holidays, because it's just in a different order, but more or less they're exactly the same. Okay, so we've got the required practicals first. So there's a few that you need to know. So microscopes has not appeared in the exams for quite a while. So understand how light microscopes work in particular compared to electron ones. So light microscopes, remember, they're the ones that are cheaper to run. They're the ones that you see in school compared to electron microscopes, whereas electron microscopes are firing electrons at the specimen. With the light microscope, we can have a specimen that's alive, whereas with the electron one, it has to be a dead specimen. So that can be a con. With the light microscope, you also need to know how to actually calibrate the machine, how to prepare the slide, and so on. So I've got a practice exam question which we'll go over for this as well. Another one that's quite an important one, so I'll just highlight the really important ones that you should definitely have in mind. So we've got microscopes, osmosis in potatoes. So remember osmosis, there's different methods you can learn for this, but osmosis is a net movement of water particles. So you can either talk about in terms of moving from dilute or to concentrated solutions, but it's better just to write water particles, high concentration to low concentration, because it's very explicit. So water particles move from a high concentration to a low concentration across a partially permeable membrane. And remember, this is a passive process. So by passive, it means it needs no energy. It just happens by itself. So osmosis in potatoes, to measure the osmosis that's actually taken place in the potatoes, you would measure the weight of the potato. And obviously, if the weight of the potato cylinder has increased, osmosis has taken place. And that's how that practical works in a nutshell. We've got enzymes and pH. Now, remember, with enzymes, we'll go into this in more detail. Enzymes work at specific temperatures and a specific pH. So, for example, if you've got an enzyme in the stomach, so remember in the stomach it's proteases, they would work at a very acidic pH because there's hydrochloric acid within the stomach. Whereas the amylase that's in our saliva, that would work at a different pH compared to the one in the stomach. So just remember, it's at specific temperatures and pH, and this investigation would test how pH affects enzyme activity. So for example, amylase breaking down into starch, but the question that they'll give you will be unseen, but it's the same idea, really. So in a previous exam question, they gave the example of pepsin, which is an enzyme you've probably never seen before, but they tell you in the question, it is a protease that's found in the stomach. So if you do end up seeing an enzyme that you've never seen before in the exam, just still think of the same practical. Don't get put off by a different name. Okay, and then we've got our food tests. So you need to know it for sugar, starch, protein, and lipids. So for sugar, it's Benedict solution. Just highlight this because it's quite important. And it would turn brick red if there was a sugar present. You've got starch or iodine, which would turn black if there's starch present. You've got protein, which is the Bioret one, and that'll turn purple if there's a protein present. And then you've got lipids. Now here I've brought ethanol, but there is another one you could do. So some of you might have learnt Sudan 3. So you can say either or. You can say Sudan-3 for lipids, or you can say ethanol. That doesn't matter. So make sure you know all your food tests. You might be given an exam question where it gives you a table and it's told you results of that food test. You have to figure out what food is present depending on the color. So make sure you've memorized all the colors and what reagent is used for each of these as well. And then we've got photosynthesis and light intensity. Now remember, with photosynthesis, you need light, otherwise there's no photosynthesis. So plants do not photosynthesize at nighttime because there's no light for the reaction to happen. And this is a pondweed experiment. So let's look at an exam question for the pondweed one, because again, this hasn't really come up that often, so it's useful to go over. Okay, so a student investigated the effect of light intensity on the rate of photosynthesis. So by light intensity, it just means if there's more light, will there be more photosynthesis? Or if there's less light, will there be less photosynthesis, and so on. So it's amount of light. So figure 1 shows some of the apparatus used. So we can see there's a stopwatch, pondweed, which is the plant that is photosynthesizing. And we've got bubbles of gas. Now, what I'd recommend, when you just jump straight into the question, try and understand what everything is in the question before jumping into the question. So for example, if we've got pondweed here, we know pondweed is carrying out photosynthesis. And you should know the equation for photosynthesis. Don't just memorize it word for word, understand it. So the plant takes in carbon dioxide from the surroundings. You then give the plant water using light. That then produces oxygen, which the plant releases, which we then use for respiration. And it also produces glucose. And that's the reason why we can eat plants, because essentially they're releasing glucose by photosynthesis. I've seen a lot of students as well, they mix symbols and words together. Try and not do that. If you either stick with all words or all symbols, and make sure it's balanced. So the symbols for this are carbon dioxide, CO2, water is H2O, oxygen is O2, glucose is C6, H12O6, and to balance that, I just need to put a 6 in front of these 3 here. So try and not mix both of them, either just learn the word version or the one with the elements in. Okay, and so if this pondweed is photosynthesizing, these bubbles of gas that the pondweed is producing surely has to be oxygen, because that's what's getting released by photosynthesis. So before actually jumping to the question, we've understood what the question's given us. And again, it's given us a stopwatch, so meaning we're measuring some form of time. All the practical questions, you should be thinking of independent variable, dependent variable, and control variable. So keep it all in your mind whilst reading the question. So name the gas produced by the pondweed in the light. Well, we've just said that's oxygen. So that's straightforward enough. Describe one way the student could change the intensity of light reaching the pondweed. Now, where students lose marks is because they list maybe one correct way and then they say something else, and then they end up losing the mark for the right answer. So if a question ever says just one and it's bolded as well, just write just one thing. Don't start writing everything, and make it as explicit as possible. So describe one way the student could change the intensity of light reaching the pondweed. Well, it's 2 marks. So describe how exactly is he putting light onto the plant. So you could use a lamp. So that's your first mark. Describe how he's actually getting light there in the first place. So you could use a lamp. And then your second mark, to change the intensity, you can just move it away from the plant for less intensity or closer. So you can just say move away or closer to the plant. And you can answer the questions in bullet points, it kind of keeps you in track as well, just to make sure you don't waffle and you're getting bang on the marks that you need to get. Okay, describe how the student could use the apparatus in figure 1 to measure the rate of photosynthesis. So whenever you see the word rate in any question, that's always in a given time. That's with any rate question. So as soon as you see the word rate, just think something per time, be it seconds, minutes, or so on. So it's saying look at figure 1, so we have to refer back to the question. So we can see bubbles are getting produced, which are in turn going to indicate that it's photosynthesizing. So oxygen getting produced would mean that the pondweed is photosynthesizing. If it wasn't getting produced, then there's no photosynthesis happening. But that's me explaining it, that's not describing it. So the examiner's report does say that a lot of people did describe what happens, explain what happens, that if there was no photosynthesis happening, then there would be no bubbles. The question's not said explain, it says describe what you would do to measure the rate of photosynthesis. So what you would say is you would count the number of bubbles. So that's your first mark out of the 2 marks. And since it's rate, you always have to say in a given time. So that's your 2 marks. You are right in saying, obviously, that there would be no bubbles if there was no photosynthesis taking place, but that's not you describing how the students are using it to measure the rate of photosynthesis. So that's 2 marks here. Let's cover some more important content to do with that. So 3 really important variables that you need to know about: independent, dependent, and control. So control is what you're keeping the same because you wouldn't want it to affect the experiment. So for example, in this question, they might ask what could be a control variable. Well, temperature would be a control variable because you don't want temperature, you're not measuring temperature and how that's affecting this rate. So for example, temperature would be a control variable in that case. You don't want it to affect what you're investigating. Dependent variable is what you're measuring, and independent variable is what you're changing. So in this case here, the independent variable, I'll just write this here. So the independent variable, so this is the one that we are changing, is the light intensity, because that's what we want to see is that having an effect on the rate of photosynthesis here. Our dependent variable is the one we're measuring, and we're measuring the amount of oxygen being produced by counting the bubbles of gas. So oxygen produced. And then control variable could be many things. One of them being temperature. For example, we don't want temperature to be this experiment. Okay, so that's the variables: independent, dependent, and control variable. Whenever you've got a 6-mark question and you're told to devise a method, make sure you state what your variables are, and also state how you would make an accurate investigation. So you would repeat the experiment and you would calculate a mean if relevant. So what I mean by if relevant, you wouldn't repeat the experiment and calculate a mean for a microscope, because what you're seeing is what you're going to see. But something like this, you would keep repeating the experiment and then calculate a mean, just to make sure there was no other influencing factors and there's no anomalies. You could even say get rid of anomalies. So these are all really important points to add when you've got a 6-marker method question. Another thing I would say as well is if they've given you a figure and they've told you certain apparatus like a stopwatch, pondweed, make sure you refer back to that in your answer as well. Go back to the question and put things from the question in your answer. Okay, we've then got aerobic respiration and photosynthesis. So we've spoken about photosynthesis. Photosynthesis needs light to work. So at nighttime, plants are not photosynthesizing. And you should notice that respiration is just photosynthesis backwards. You can see these two, exactly the same as these two in aerobic respiration, the products. And in aerobic respiration, the reactants are the same as the products in photosynthesis. So if you remember one way, you should remember the other way. Now, one thing just want to say about respiration, the whole purpose of respiration is to release energy. and we need energy to be able to do everything. So that's the whole purpose of aerobic respiration. A lot of people say produce energy. You can't say the word produce, because if you think back to physics, the law of conservation of energy, we can't create or destroy energy. So therefore, respiration just releases energy. And another thing about respiration as well, it's not breathing. So if you ask them the question, what is respiration? You just say it's the process. So this equation here, you can put that equation down, and it's the process which releases energy. Yeah, make sure you say releases. That's really important. Okay, so aerobic respiration means with enough oxygen. Now, if there wasn't enough oxygen, then anaerobic respiration would take place. Now, remember, anaerobic respiration is different for both an animal cell and a plant cell. So in an animal cell, so just like us as well, lactic acid would get produced. And that lactic acid, that's what causes the stitch feeling. In plants, it's known as fermentation because ethanol gets produced. So make sure you know the two differences with anaerobic respiration. But generically, when you're speaking about respiration, you're speaking about aerobic respiration. Okay, so what three extra organelles do plant cells have compared to animal cells? So the extras that plant cells have are chloroplast, cell wall, and permanent vacuole. Make sure you know the process for each of them. Chloroplast is where the photosynthesis happens. The cell wall, this is what's providing extra protection to the plant cell outside. And remember, it's made up of a sugar called cellulose as well. The permanent vacuole, this contains cell sap, so nutrients for the plant cell, but also another thing it does, it's quite big in the center of the plant cell, and it pushes all the chloroplasts to the edge of the cell. Now, at the chloroplast, photosynthesis happens, and photosynthesis needs light. So the reason why it pushes it to the edge of the cell is so it can get the maximum amount of light. So that's the reason why all the palisade cells as well, if you look on top of a leaf, I'll just quickly show you. So if you look on the leaf, you can see the palisade mesophyll. So these are just all the palisade cells here. They're on the top of the leaf because they want to get the maximum amount of sunlight so photosynthesis can take place. Okay, methods of transport. So make sure you know what diffusion, osmosis, and active transport is. I'd say the most important one for GCSE is diffusion. So it's movement of particles from a high concentration to a low concentration, and it's passive, which means no energy. Sometimes you'll just say it moves down a concentration gradient. You might also see that as well. Down a concentration gradient. So what a concentration gradient means is just high to low concentration. So it just happens by itself. If I had some perfume and I sprayed it, naturally that perfume will spread to areas of low concentration, just because that's a natural process. So that's what diffusion is. Diffusion is how oxygen moves from the alveoli into the blood, and how glucose gets into our cells and so on. So it's the most common process within the body. It just happens naturally. Osmosis is the same as diffusion, as we spoke about the potatoes experiment before, but this time it's water particles. Again, high to low concentration, and it moves through a partially permeable membrane. And again, that's passive. So it goes down a concentration gradient as well. So both osmosis and diffusion go down a concentration gradient. Active transport, this is the one that's the opposite. It goes from a low concentration, so let's say 3 particles, to a high concentration. So we say this is going against a concentration gradient. So that's going against the concentration gradient because it's not natural to go from low to high. So since it's not natural to move particles where there's less to somewhere where there's more, this process needs energy. And where does it get that energy from? Respiration. So everything you will speak about in Biology Paper 1 always links back to respiration somehow. Okay, another really important thing that tends to come up almost every single year is understanding how enzymes work. So I think the most important thing, first understand what is an enzyme. Well, it's a biological catalyst. What does a biological catalyst mean? It means it speeds up a reaction without getting used up, which means it can be used again and again and again. And it's specific. So certain enzymes will break down certain food molecules. So there's 3 that you need to know about: amylase, protease, and lipase. Sometimes you might see amylase called carbohydrates. You can write amylase or carbohydrates in the exam, you'll get the same mark, they're the same thing. Amylase breaks down starch, and it breaks down starch into glucose. Again, you could say carbohydrates as well instead of amylase. So that breaks it down into glucose. We've got protease. It's got in the name. Protease breaks down proteins. And what does it break down proteins into? It breaks them down into amino acids. And finally, lipase, they break down lipids. Try and say the word lipids. Don't say the word fats, even though lipids are fats, but they want the specific word lipids. And it breaks down lipids into fatty acids and glycerol. You have to say both of these. So the whole purpose of enzymes is breaking down big molecules into smaller molecules that can get absorbed by the body. So we can use the glucose, we can use the amino acids, and we can use fatty acids and glycerol. That's the whole purpose. You need to know where these enzymes are also found. So amylase is within the saliva. Protease is in the stomach. So that's in, I would say, salivary gland. A common mistake students make, they say it's in the mouth. You can't say it's in the mouth. You have to be really specific that it comes from the salivary gland. Protease, they found in the stomach. So where the hydrochloric acid is, so they're well adapted to them. And you've got lipase, which they found, it could be found in the pancreas, it could be found in the small intestine. All 3 of these are found in the small intestine and the pancreas. But the extra specific ones, salivary gland and stomach, amylase and protease is what you need to know. Okay, they're temperature and pH dependent. So you should know that the body temperature is 37 degrees Celsius. And if you move away from that temperature, the enzymes denature, which we'll come on to. pH as well, specific enzymes will work at a particular pH. So proteases work well in the stomach at a low pH, since the stomach is acidic. Remember, your pH is between 0 and 14, whereas where 0 to 3 is really acidic, and then 12 to 14 is really alkaline. So pH specific as well. Your salivary glands are not acidic, so the amylase would be suited to a different pH there. You need to know what the graphs look like, which I'll go over as well. But before going over that, let's talk about the lock and key model and how the enzyme actually works. So the lock and key model says, okay, so literally a lock and key. So as you can see from the first picture, the purple enzyme acts as your lock, and then the substrate is the key that fits inside that lock. Your substrate has to fit into the active site. So the active site is the shape on the enzyme where the substrate is binding to, and that's what makes it specific. So we said all enzymes are specific to certain molecules of starch, proteins, lipids. That's the reason why, because they've got a specific active site. And it's when this active site denatures, so it changes shape, is what causes the issue. So let's talk about pH and temperature affecting it. If you do not have a suited temperature pH, so let's say we're at 39 degrees Celsius, your enzyme will denature. In the exam, you can't just say the word denature. You have to give a bit more explanation. The enzyme denaturing means the active site has changed shape. So active site has changed shape. That's really important to mention. So if the active site has changed shape, then the enzymes are no longer functional because it can't take the substrate on. The shape has been changed, so then therefore it can't get broken down. Okay, just want to speak about the diagrams quickly about enzymes and just add it to here. So with enzyme, so temperature and pH, your temperature graphs tend to look like this. So let's just say this is temperature, so it's increasing as you're going along the x-axis, and then we've got the rate of reaction, so the amount of substrate that's getting broken down. So at first, when you increase the temperature, the rate of breaking down the molecules into smaller molecules increases, and then it gets to a point where it just drops. Let me just make that a bit more clearer. So it increases, and then it drops at a particular point. And that particular point is after 37 degrees Celsius. So make sure you remember that sweet spot, 37 degrees Celsius, which is what our body works at. After this point, you might be told, the question might say, explain what's happened here. So we call this the optimum temperature. It's the best temperature for the enzymes. So what's happened? The enzymes denatured, the active site has changed shape. So therefore, no substrate can be broken down. You have to be really specific in your points. So they're your 3 points. What's happened after that? Same goes for pH. Now with your pH graphs, the way they look, depending on where they're found, they just follow a shape just like this. So say, for example, this is for a protease enzyme. So this is the pH level. This is, again, rate on the y-axis. So let's say between here is 0 and, let's say 4 there. And then after 4, it just drops down. The reason why it drops down is because it's not suited to that pH. So the peak, optimum peak is basically the optimum pH for that enzyme. So if you see an enzyme that you've never recognized before, recognize the shape. The top peak is the optimum. That's the best pH it works at. They might ask you what's the optimum pH. Find the peak and then just read down, and then that's your answer. One other thing I want to mention with graphs, if you've got a graph and you're told to read off, if you want to work out the value of one box, so let's say 1, 2, 3, 4, 5, let's say that's 0 and 10. If I want to work out the value of one box as I'm going up, you're just doing 10, so that's 10 minus 0, so the space between there, divided by the number of boxes in between, which is 5. So 10 divided by 5, that's just 2. So the value of one box is just 2. So 2, 4, 6, 8, 10. So that's how you find the value of one box when you're trying to read off graphs in biology, especially. Okay, let's go back up. Okay, so we got on, you should recognize this picture. So here we've got alveolus. We say alveoli when we've got many. And this is a capillary here. So remember the whole purpose of the respiratory system, we breathe in through our mouth and our nose. The oxygen goes down our trachea, which splits off into the bronchi and then the bronchioles. At the end of every bronchiole is the alveoli. Just quickly show you a diagram of that. To make sure you know that structure there as well. And then at the end, we've got the alveoli. How's the alveoli suited for what it does? So it's got the oxygen there at that point. There's loads of them. So meaning that they've got a very large surface area. That tends to be a reason for almost everything as well in biology. If they ever say, how's so-and-so adapted for carrying out its process, large surface area tends to be the answer for that. Here we can also say as well, the alveoli has very thin walls, and so does a capillary. It also has very thin walls as well, because diffusion's happening between them. So diffusion is taking place. And remember, when diffusion's taking place, I know we do speak about oxygen quite a lot, like the oxygen's coming out of the alveoli into the red blood cells. But at the same time, carbon dioxide is also leaving the capillary through the alveoli that we then breathe out. Probably won't take a break just because it's only going on till 6:30. But if you do want to take a break, this is recorded so you can watch it back. as well. Okay, so the alveoli, how is it adapted for gas exchange? Well, we've got large surface areas I spoke about, thin walls, so that means like a short diffusion pathway, so oxygen can go in and out really easily, and it's got a good blood supply. Now, the reason why good blood supply is important, this blood has to constantly keep moving because we need blood that doesn't have oxygen in, so then the oxygen can come into the capillary. So for example, oxygen's got in at this point, this blood needs to constantly move around so this new deoxygenated blood can come back around so the process can still keep happening. So that's the reason why good blood supply contributes to it as well. Make sure you know the order of organization. Cells, loads of cells together make a tissue, loads of tissues together make an organ, and them organs together give an organ system. So you've got the digestive system. In the digestive system, we've got our small intestine, large intestine, stomach, pancreas, and so on. Circulatory system, we've got the heart, we've got all the blood vessels, and so on. So different organ systems made up of different things. Okay, we've got another exam question here. So it says here, what is the process? What's this question related to? Oh, okay. So what's the process by which water is lost from the leaves of a plant? Always use process of elimination if you can. So it's not osmosis, because osmosis is the movement of water particles from a high to a low concentration. So that's not water lost from the plant. It can't be photosynthesis because we've just discussed what photosynthesis is. So therefore it's transpiration. So transpiration is water lost from the leaves of the plant. And there's so many different factors that can affect this. Transpiration tends to come up quite often. So I would recommend really going over transpiration. These are the factors that affect transpiration. It's not osmosis, because osmosis is the movement of water particles from a high concentration to a low concentration through a semi-permeable membrane. This water is not lost through a semi-permeable membrane. It can be lost by evaporation because of the light intensity. It could be lost because maybe you've not watered the plant enough as well, for example. Maybe there's too much photosynthesis happening because the temperature is quite high. So it's not osmosis, it's transpiration. So make sure you understand each factor affecting transpiration. So you've got humidity. What does humidity mean? Humidity means the amount of water particles are in the air. So you could say the UK is quite a humid country because we're surrounded by quite a lot of ocean, meaning we've got so much water particles already in the air that water is not going to leave the plant because it's high concentration outside. There's no concentration gradient. That's why that affects it. Air movement, so if it's quite windy outside, the wind will blow out the water particles off the plant. Light intensity, the more light, the more photosynthesis, more water being used. Water availability, meaning watering the plant, they have to get the water from somewhere, or potentially rain. Rate of photosynthesis, again, ties in with light intensity, and temperature as well. Okay, stomata and guard cells, these are at the bottom of the leaf. I'll just show you the leaf structure here. You can see right on the bottom there, we've got the stomata. They say stoma if it's just one, and the guard cells control the stomata opening and closing. So why do we have stomata? It's for gas exchange. It's to allow gases into the leaf. The guard cells control the opening and closing of that. Make sure you understand these two words, turgid and flaccid. So if you say the plant cell is turgid, it means it's full of water. So if a plant's full of water, you would consider it healthy, so it's ready for photosynthesis, because remember, for photosynthesis, we need water. Find the equation here. You can see here, for photosynthesis, we need water. So if there's no water, there's no photosynthesis. So when a plant is, the cell is turgid and full of water, we can then open the guard cells. The guard cells will open the stomata to allow for gas exchange, so the oxygen and carbon dioxide in and out. If the plant cell is flaccid, it means it's lost water. So maybe it might be, the plant might be on a desert, for example, so it's lost so much water. So therefore the plant's dying. So the guard cells will then close the stomata to prevent water from leaving the plant. Another thing to remember about guard cells is they're light dependent. light dependent or light sensitive? Light sensitive is what I would say. So the guard cells are essentially guarding the stomata, is what you can think. They're light sensitive because at nighttime they will close the stoma just to make sure it doesn't lose water at nighttime, and it conserves it because the plant's not the plant is not photosynthesizing at nighttime. That's another point I want to make as well. In a plant, both photosynthesis and respiration takes place. So a plant respires and photosynthesizes. An animal cell just carries out respiration. Just think of it as a human. As humans, we don't carry out photosynthesis. We don't have plant cells. So make sure you understand that as well. Okay, let's move on to types of pathogens. It's been a while since malaria has been tested in the exam. So have a good idea of what malaria is and what it's caused by. The mosquitoes are not the actual issue. The issue is the mosquito carrying the protist. So let's go over them. You've got four different types of pathogens. So you've got bacteria, virus, fungi, and protist. There's certain examples that you do need to memorize. So if I just go to the summary sheet. So for example, with bacteria, salmonella, food poisoning, if you're not cooking chicken well enough, for example, and you've left that bacteria on, that would cause salmonella. Make sure you know the symptoms associated with that. So salmonella can cause a fever, it can cause vomiting, it can cause diarrhea. A virus could be measles, HIV, and so on. Fungi could be athlete's foot. You've got a rose black spot on plants. So what rose black spot causes is the leaves to lose its green color. So if the green color is lost from the plant, then the plant won't photosynthesize and in turn die. And then you've got protist, which we'll talk about in this. So a protist is it's just a single-celled organism. It's just a eukaryotic cell. You don't really need to know much about it, but it's a cell that's carried by a vector. So it's a cell, and it's carried by a vector. And this vector is basically the mosquito. Mosquito in the example for malaria. So the mosquito gets a protist from somewhere, it carries it, and then it bites the human, and it passes that protist on. So that's how malaria is caused. Now it says here, scientists are developing a vaccine against malaria. Suggests how a vaccine against malaria could reduce the spread of disease. Anytime you've got a question that has vaccine inside it, you're talking about immunity. So you're talking about immunity. So that's the first point you'll make. So you're making people immune to this disease. So you're not actually describing what the vaccine's doing. I'd say that's one big tip for tomorrow's exam. Whenever you've got a question like suggest how a vaccine against malaria could reduce the spread of disease, you're not saying what the vaccine's doing. You're not describing that you're injecting a weakened or dead form of the pathogen and then the antibodies getting released and so on. You're just explaining how is it reducing the spread of the disease. So please try and answer the question. Don't waffle and write so much because you lose marks for that waffle. Okay, so we've made people immune. So what has that done to reduce the spread of disease? It means that fewer people to pass the pathogen onto. Because if more people are immune, then it's going to spread less around. So that's what it's referring to, and that's 2 marks. I would recommend using bullet points in the exam just so you've got the 2 marks. Always look at every single mark for each question. If it's 2 marks, you need 2 points. If it's 6 marks, you want to get in a good structure. 1 mark, just write just one thing. Here it said give one way. It's very specific to just give one way of controlling the spread of malaria. And it says do not refer to vaccine in your answer. So because they've already told you vaccine, so they don't want you to use that. Okay. So it says, do not refer to vaccines. There's quite a few different things you can write here, but just write one. So you could say draining swamps, because that's where the mosquitoes go to go get the protist. So you could say draining swamps. You could say using mosquito nets just to prevent the mosquito from biting you. You could say kill the mosquitoes. So kill the mosquitoes because it means they won't be a vector. Okay, we can say insecticides because that again kills the mosquito. Anti-malaria tablets, I probably wouldn't write that because you want to prevent it from happening. What else has it got? Yeah, and that's it. Make sure you don't write anything like antibiotics or anything like that. Remember, antibiotics only cure bacteria and nothing else. Also, I've already wrote that here. So antibiotics only cure bacterial infections and not viruses, not fungi, not protists. So don't write that for GCSE. Okay, let's move on. We've got the circulatory system. Hopefully you do end up getting a 6-mark question on this tomorrow because it's quite a good 6-marker to get. So make sure you know the whole process of the circulatory system. And I wouldn't recommend just memorizing it, like actually understand what's happening. The whole purpose of the circulatory system is to transport blood around the body. Now, the reason why it's transporting blood around the body is because we need to go give oxygen around the body. In that blood will be oxygen in the red blood cells in particular. That's going to be transported all around the body, so it could be used by our cells for respiration. And why do we respire? For energy. So to release energy. And so you can see it just follows all the way through. So that's the whole process of the circulatory system, why we do it. Let's talk about the actual process. So we've got blood vessels here. We'll start off with A. So at A, the blood has come from the body. So there's 3 things you should always know about each section to do with the heart. Know where it's coming from or going to. Know whether the blood is oxygenated or deoxygenated. And the name of the blood vessel. That's the 3 things you need to know about each section. Let me just get rid of this so I can put this clearer. So at first, it's coming from the body. And the reason why it's come from the body to the heart is because the blood is deoxygenated. And the reason why the blood is deoxygenated is because it's used all the oxygen in the blood for respiration. So it's coming back to the heart to get more oxygen. So that's the whole purpose. One thing I want to mention just before going into the blood vessels, remember there's 3 types of blood vessels. You've got artery, you've got a vein, and you've got a capillary. Think of blood vessels like roads in the body. They don't end, they're all joined together. But depending on which direction it's going in, depends on whether it's an artery, vein, and the capillary joins them 2 together. So artery takes blood away from the heart. And the way you can remember that is the A in front. So artery takes blood away from the heart. For the vein, it takes it to the heart, so just the opposite. And the capillaries is what joins these together, and it's also where the gas exchange will happen. So how can I describe it? I'll just say gas exchange takes place here. And the reason why it does is because it's 1 cell thick. Okay, so capillary is what the alveoli is wrapped in. Arteries and veins, just remember direction. So if at first here, blood is coming from the body to the heart, since it's coming to the heart, it's a vein. And the type of vein it is a vena cava. Okay, so that's the first step. It then passes through the right atrium and then goes down the valve. Now, the whole purpose of the valve is to prevent backflow of blood. Prevent backflow of blood. You basically want the blood to go in 1 direction. Go in 1 direction. So if they ask you what's the purpose of the valve, prevent backflow of the blood and make it stay in one direction. Okay, it then passes through to the right ventricle, sorry, and then it passes out this blood vessel here. Now, where's it going now? Well, it's going to the lungs. And the reason why it's going to the lungs is, to the lungs. So the reason why it's going to the lungs is because it's going to go get oxygen. Tricuspid valve and bicuspid valve. You don't need to know that for AQA. I'm not too sure if you're doing a different exam board, but for AQA, you don't need to know the actual type of valve for that. You just call it valve. Anyways, as I was saying, it's going to the lungs to go get oxygen. So it's still deoxygenated at this point. So it's still deoxygenated. And whenever, since it's going away from the heart, it's an artery. So you can see it's going away from the heart. So it's an artery. And since it's going to the lungs, we call it a particular name. We put pulmonary in front of it. So anytime you see the word pulmonary, it's something to do with the lungs. So that's the easier way to remember it. It's going away from the heart. It's going to the lungs. So therefore, blood vessel B is the pulmonary artery. Okay, so now it's gone to the lungs. And it's gone to the lungs to go do this, this diagram here. So it's gone to the lungs. Here's the capillary, which will join to that pulmonary artery. And here is where the gas exchange happening. So oxygen's coming into the blood, and then it's going to come back to the heart. And remember that process takes place with diffusion. Okay, so now the blood has got oxygen inside it. So let's come to D now. And this is an actual heart diagram as well from the exam for AQA. So we're going to come over to D now, the left atrium. So it's come from the lungs. And this time it's got oxygen inside it because it's already got that oxygen. So we say it's oxygenated. And since it's coming to the heart, it's a vein this time because it's coming to the heart. And again, since it's, just write this here. Since it's come from the lungs, it's a pulmonary vein. Okay, and then it then passes through the valve again to prevent the backflow, and then to the left ventricle, and then it's coming out of vessel C, and it's going to the rest of the body. So to the rest of the body. And the reason why it's going to the rest of the body is because it's giving that oxygen to all the cells around the body so it can use it for respiration. So it's oxygenated to go give it to the rest of the body. And since it's leaving the heart, it's going away from the heart, it's a type of artery and it's got a special name. It's called the aorta. And that's the circulatory system. So then the body will use it for respiration and then it'll come back to the heart and the whole cycle starts again. Few things to know about this. You need to know about pacemaker cells. The pacemaker cells are found in the right atrium. And the way to remember that, if you forget that they're in the right atrium, think that the whole process starts in the right atrium. So if there was, the pacemaker cells have to be in the chamber where the process starts. So that's how you can remember it's the right atrium. The pacemaker cells are basically giving that electrical impulse to the heart to get it to contract and start. Some people don't have pacemaker cells, so they can get an artificial pacemaker put in to the right atrium for that as well. Another thing about the circulatory system, they call it a double circulatory system. The reason why they call it a double circulatory system is because blood enters the heart twice. So blood enters the heart twice in every cycle. So you could see it enters the heart here at A. First, it's entering as deoxygenated, and then it enters again the left atrium once it's got oxygen, and then it's giving it around. And that's happening every second, every heartbeat, this process is happening. Okay, if you get any coronary heart diseases, the way we resolve them is stents and statins. Statins are tablets. Stents are little tubes or pegs that they stick inside the vessels to open them back up again. So just imagine you've got a vessel, and I'll just do it on the bottom. So I'll just write coronary heart disease. So the word coronary is to do with the heart. If you see the word coronary. So coronary vessels are vessels in the heart. Pulmonary is to do with the lungs. Okay, so if you get coronary heart disease, it means your vessels most likely may be blocked. So let's say it's blocked with fatty deposits. So if you get fatty deposits on your vessel walls, you're making it smaller. So if you're making a vessel smaller, you're increasing the flow pressure flow of blood through that vessel, so it's going much faster. And if it's going faster, it means increased heart rate and high blood pressure, and then that can lead to a heart attack. It can get worse if you keep maybe a poor diet and you keep having more fatty deposits, and it completely blocks one of the vessels. As soon as you get that blockage, no more blood can pass through, and if blood cannot pass through, then that whole process we've just spoken about can't take place. So therefore, that's also what causes a heart attack. One other thing to mention about the heart as well, I've seen this as a past exam question. They can say what's the differences between the artery and vein. So remember with the, I'll just show you a quick diagram here. So we can see here, the artery has a smaller lumen. Now, when we say lumen, that just refers to the hole in the blood vessel. So it's got a smaller lumen, and the vein's got a large lumen, so a bigger hole. The reason why is arteries are taking blood away from the heart. So since it's taking it away from the heart, they need to be at a really high pressure. So if you imagine you've got like a hose pipe, if you squeeze it and you make it smaller, the water will go through it a lot faster. Same idea here. Because it's smaller and it's got thicker walls, the pressure is going to be a lot faster through the artery. Now with the vein, the pressure is not that high, the blood going through, but we still need the blood to go through in one direction. That's the reason why the vein has a valve. So it keeps the blood in one direction. So if you ever get asked the question tomorrow that why does the vein have a valve but the arteries don't, is because the arteries are already under high pressure to keep the blood in one direction, whereas the vein has blood passing through it at a low pressure, so the valve needs to keep it going in one direction. Okay, and then capillaries are just one cell thick. Okay, let's find another exam question. Okay, pacemaker cells found in the right atrium, which we've already discussed. Okay, let's see this question. If you've got any questions related to any of this as well, ask in the chat. If not, we'll have some time at the end as well to go over some questions as well. Okay, so it says here beta blockers are another type of drug that slows the heart rate. So you've not really learned about beta blockers, but again, they can just ask anything in the exam. So you just need to be prepared for anything. Don't get worried that you've not seen the word beta blockers, but you know it's a drug that's slowing the heart rate. So in your mind, just think, okay, if I slow the heart rate, I'm slowing the whole process of blood getting pumped around the body. So hopefully, Akib, you'll get 6 marks after this. So yeah, if I'm slowing the heart rate around the body, then blood is not getting around the body as fast as possible. And if blood is not getting around the body as fast as possible, that means less oxygen is getting around the body. And if less oxygen is getting around the body, less respiration is taking place. And if there's less respiration, then less energy being released. So you get more tired and fatigued. So just try and think of all this as you're reading questions. Don't just blindly read them tomorrow. There's always a reason for telling you something, and that's a big clue what they've told you here. Table 2 shows some information for people who do not take beta blockers and for people who do take it. So do not take and who do take it. Stroke volume is the volume of blood pumped out of the heart each time it beats. Okay, so stroke volume is the volume of blood pumped out of the heart. So okay, we'll look at the table and see what happens after. Cardiac output is the total volume of blood pumped out of the heart each minute. Okay, let's have a look at the table and understand it. So we can see here we've got no beta blockers taken for this one. So this is just for a normal person. And this is for someone who's taking beta blockers. So this means this one's going to have a slower heart rate. So I would highly recommend just annotating the question before you just jump straight into answering the questions. And let's just quickly look at the numbers. We can see the heart rate in beats per minute. So we can see it's getting lower for the person who's got beta blockers, which is expected. They're going to have a slower heart rate. Okay, that's expected. Stroke, and we can see it's at rest and during exercise. So I'm going to compare that at rest. We can see the stroke volume. Well, we can't compare that because there's an X here. But cardiac output, we can see is decreased for beta blockers. So really, overall, everything has decreased for the beta blockers as expected. It says here some people who take beta blockers get out of breath in the exercise. That's because they're not aerobically respiring because there's not enough oxygen. So that's what's happening. Explain why beta blockers can have this effect during exercise. You should refer to the information given in table 2. So everything we've just basically discussed before even reading what the question is what the answer is to this. And the most important thing is referring to the information in table 2. There's a reason they've given you it. This X value here, I think I missed the question on that and what that should be. Okay, let's just do this question anyway. The 6 marker is irrelevant, that X, to be honest, to this question. So explain why beta blockers can have this effect during exercise. So let's explain the effect that beta blockers are having first. I'd recommend doing them in bullet points. You can write a paragraph if you want, but bullet points just explicitly tells the examiner, this is the point I'm making. So you would say beta blockers slow down heart rate. So you would say less blood pumped around the body in a given time. At the same time, we should also refer to the table actually as well. So if I move this down, I could give a point for this one here. So I've said beta blockers slow down heart rate. Let's refer to figure 2. And it does the heart rate. So you can say heart rate, so this line here, for no beta blockers given, no beta blockers. Let's just say during exercise, because they care about exercise. We can say during exercise is 150, and then you can say whereas for, so you can say whereas for taking beta blockers is 88. So you're basically proving the point you're making. It says here beta blockers slow down heart rate. They've told us that right in the beginning of the question. You're then referring to the table to back your point. So really the answer is already given in the question. Okay, so less blood is then pumped around the body in a given time. Again, you can give an example from the table. So it says here stroke volume is or we could just say cardiac output. You could use any of them, you'll still get the mark. So cardiac output is the total volume of blood pumped out of the heart each minute. So let's talk about cardiac output. So we can say cardiac output for the I'm just going to write BB for beta blockers. So we can say cardiac output for no beta blockers during exercising is 18,000. And for taking beta blockers is 8,624. So that proves that it's less blood pumped around the body. So you're not just saying a statement without backing it up. Okay, so that's about the cardiac output. What else? So Some people explain why beta blockers have this. Okay, so why they get out of breath. So if less blood is pumped around the body, that means less oxygen is getting around the body. If you want to be specific that the oxygen goes into the red blood cells, you don't have to, you don't get a mark really for it, but because this is a 6 marker, there's a leeway for whatever you write, to be fair. Less oxygen is getting around the body, so that means you could say oxygen is needed for respiration. You can talk about respiration. I would even put the equation down. You could say oxygen, so what's the respiration equation? You could say oxygen, so you breathe in oxygen, we get glucose from our food, we then release carbon dioxide, I'm just going to write this, plus water. Make sure you don't mix up symbols and words up, just doing that in essence of time. You can then say respiration releases energy. Now, if you're not releasing energy, then you're going to get out of breath quickly. Out of breath if not respiring. And we've explained it. So why does it have this effect when they exercise? You're getting out of breath because you're not respiring, so you're not releasing, because you're not releasing energy. Okay, and I would say that's a good 6 marks there. And we've also referred to the table as well. So let me just double check what's also on the mark scheme. Rate of respiration increases during exercise. Yeah, so you can say that you're, you could talk more about what's happening during exercising. You need more oxygen during exercising, right? Yeah, that's all covered. Then it says you must refer to obviously information in table 2. They want you to give figures for that. What was the value of X in the table? I think that was the previous question where it said what is X, but I'm missing the question here. At the end of this session, Akib, I'll have a look and then we'll come back to what that value of X is. I think I've missed out the question when making this. Right, let's continue. Hopefully that did make sense. How much have we got left? And we've got like 10 minutes left. Okay, let's quickly go over this last one. So this 5 markers, quite a good one. Now on the summary sheet, not too sure how many of you guys have had a look at that summary sheet. There's a section about surface area to volume ratio. So we've talked about how large surface area is good in contributing to like faster absorption of things. So places where it happens, we've got loads of alveoli in our lungs, we've got loads of small ones, and rather than one big alveoli where gas exchange takes place, because we want to absorb oxygen in and out at a fast rate. We've got villi which line the small intestine, and that's to allow for greater absorption of glucose into the blood and so on. Okay, so we've got a question here about large surface area to volume ratio. It says here cube X represents a bacterial cell, so that's just a bacterial cell which is quite small, and cube Y represents a small multicellular organism. So for example, like a human or I don't know, a cat or something. Okay, so we can see it's vastly bigger compared to this one here. Okay, it says a bacterial cell can absorb all the nutrients it needs by diffusion through its outer surface as expected. For example, the alveoli can, but I'm not going to be absorbing oxygen through my skin if I was cube Y, because I'm too big. Explain why a multicellular organism cannot absorb all the nutrients it needs by diffusion through its outer surface. You must include calculations in your answer. So a big clue they've given you here. We're going to be working out surface area to volume ratio. So as soon as you start, you see a 3D shape, they'll only give you a cube, hopefully. They don't give you something really strange, but if they do, you're just working out surface area and volume. If they give you a sphere, for example, which I hope they don't, they should give you the equation for finding out the surface area of a sphere and volume of it as well, or a cylinder and so on. But usually it is cubes because it's easier to work with. So we're working out the surface area to volume ratios. And we'll explain the answers that we get. Okay, so let's work it out for cube x. Let me just keep this here. So that's what it's referring to. Okay, so for cube x, so remember it's a cube, so all the sides are exactly the same. So for the surface area, I'm going to find the area of one face and I'm going to times it by all the other faces. Now remember, cubes have 6 faces. So I'm going to find the area of one face and I'm going to times that by 6. So the area of one face is just 0.0002 times 0.0002. We're just squaring it to find the area of that front face. Write down all your working out as well. Again, it's method marks, just looking on the mark scheme. 0.0002. Let me just work down the calculator. I'm guessing I'll get something in standard form. Times 0.0002. Okay, we get 4 times 10 to the minus 8. Don't worry about converting the standard form, just leave it as is. But that's not our final answer. We've worked out the area of one face. We then need to times that by 6 because we've got 6 faces. So I'm going to do 4 times 10 to the minus 8 times 6, and that gives me 2.4 times 10 to the minus 7. Okay, so I found the surface area. I need to find the volume now. So the volume is, we just do length times height and then the width of it. So we're going to do this times this times this. So I'm doing 0.002 times itself 3 times. So we can just say cubed basically. So 0.0002 cubed, that gives me 8 times 10 to the minus 12. So that's my surface area to volume ratio. It's 2.4 times 10 to the minus 7 to 8 times 10 to the minus 12. Okay, so we worked out for X. Now let's work out for Y. So these were the bacterial cells. So for cube Y. So again, I'll just do this a bit quicker. So surface area, I'm going to do 11 times 11 to find the area of the first face, and then I'm going to times that by 6. So 11 times 11, and then I'm going to times that by 6. So 121 times 6 gives me 726. And then the volume is just 11 cubes. So 11 times 11 times 11. We times our centimeter like 3 times the volume. 11 cubed, which gives me 1,331. So our surface area to volume ratio here is 726 to 1,331. Okay, so to compare both of these, we need one of them to be the same. So I'm going to make the volume part change to 1. So it's always better just to do your volume. You can just compare that then. So we need to change both of them to 1. Now here, if I want to change this to 1, I'll do this in a different color. I'm going to divide by 1,331. And remember with ratios, whatever I do to one side, I do to the other side. So I'm dividing that by 1,331. And I get a 1 here. And then 726 divided by 1,331. I get 0.545454. I'll just round it to 2 decimal places because we're comparing. So 0.55. And then we're going to do the same here. So I'm going to get the right-hand side to be 1 so I can compare it to the bottom ratio. So I'm dividing by 8 times 10 to the minus 12. So I get 1 for this side. So I have to divide the other side as well by 8 times 10 to the minus 12. And I get over 8 times 10 to the power minus 12. I get 30,000. Okay, so now we just need to explain our results. So we've got 30,000 to 1 for the smaller one as expected. I expected a really big surface area compared to the volume. So we've got a large surface area for cube X as expected. And we've got a small surface area for cube Y. So that's all you're describing. So you can say, there's a few different things you can say here. You can say cube. Let me just put this in a different color. I'll put my explanation here. So cube X has a larger surface area. So that allows it to absorb nutrients. So you say cube Y, vice versa, has a smaller one. You would also say the diffusion distance is shorter for cube Y. Not Y, sorry, X. It's shorter, so because it's shorter, diffusion can happen through it. What else could you say? Diffusion distance is longer, volume supplied, that's the same thing. Diffusion rate per unit volume is slower in the multicellular organism, which we can see. The diffusion rate for the multicellular organism, so Y, is much slower since these two numbers are much more closer together compared to cube X. So that was quite a tricky one, but just the calculations alone will get you really high marks anyway. And always expect a certain answer as well. You expected the surface area to be lower for cube Y compared to cube X. And that's that one there. Just for the last 5 minutes, I just want to quickly speak about these two questions here. I've already wrote the mark schemes here. So the question says a microscope has a 5 times eyepiece lens. So they've told you this in the question. Describe how to use this microscope to observe a prepared slide of root hair cells at a magnification of 50. So in the question, they've already told you the root hair cells are prepared already. So if you wrote anything about how to prepare the slide, you wouldn't have got any marks because you can just see it just says place. This is the mark scheme. It already says just place a slide on the stage. So that's where a lot of people lost marks with this one. It also says in the question at a magnification of 50. So if we've got a times 5 eyepiece lens already, well, we need the objective lens of something to give us 50 altogether. Now remember, our two magnifications, we times them with each other. So 5 times something gives me 50, and that's 10. So this 10 would be the objective lens. So that's the reason why they've told you this in the question. They want you to be really specific with that calculation. You can see on the mark scheme, it says here times 5 eyepiece and times 10 objective lens. And then it's just a normal method. Always start off with the lower lens first because you've got a wider view of your specimen, and then slowly keep focusing down and increasing magnification. Okay, that's there. Yeah, that one's fine. Okay, so the mark scheme's there. And just quickly speaking about antibiotic resistance. Antibiotic resistance is, let me just explain it here, and then wrap it up there. So let's say, remember antibiotics are just for bacterial infections. Let's say someone is poorly and they're not getting better, so they've gone to the doctors and the doctor's giving them some antibiotics. They prescribe them some antibiotics to complete for 7 days, let's say, for example. Now they've told them to complete the whole course, like even if you get better, take them for the full 7 days. So the person's taking them for 7 days and they've noticed, like, over time, so let's say we're on day 3, 3 of them have, let's just say 3 of these bacteria have gone. So the person's feeling a lot better. So then let's say they stopped taking the antibiotics. So they stopped taking antibiotics because they thought they're better, so they don't need to take them. What then happens is these bacteria that are still left behind, they start mutating. So when they start mutating, they start changing and they get more aggressive and they stop working with antibiotic and they'll replicate. And now that person's become poorly again, but then they can't have antibiotics again because they're now resistant to it. So that's what antibiotic resistance is here. So I've done a practice exam question there for you to have a read over. And so explain how the overuse and misuse of antibiotics can lead to antibiotic resistance. There's a mark scheme answer, what I've just described. Part B, describe 2 ways to reduce the development of antibiotic-resistant bacteria. So reducing the use of antibiotics when not necessary. That's the reason why you can't go to the doctors, you can't go to the pharmacy, and ask for antibiotics. Your doctor has to prescribe them because they have to see if it's worth giving to you, or whether it's just worth you just getting better by yourself, naturally. And then the second point is completing the full course of antibiotics to ensure all bacteria is killed. So yeah, just as I described below, and we'll wrap it up there. Hope that was helpful. Has anyone got any questions on Biology Paper 1 in general? Anything that you're quite nervous about, or topics that you're confused with? For the X question, the table, the cardiac output equals stroke volume times heart rate. Oh, okay, was that given in the question? So I probably missed that part out. Okay, so for X, if the cardiac output, which is this, equals stroke volume times heart rate, so this times this gives you this. Okay, so then to find X, would you just do 2,800 divided by 52? I haven't got the mark scheme with me for that X there, so. Okay, any other questions related to anything else in Biology Paper 1? I couldn't obviously go through everything, but I've tried to go through as much as possible. Another thing I've not gone through that I would recommend going through is the cell cycle. So the fact that DNA replicates first, then mitosis happens and it splits off, and then the last stage, which you can call cytokinesis, but you don't have to, but it's when the nucleus divides. Make sure you know them 3 stages to the cell cycle as well. But if there's not any more questions, we can wrap it up there. I'll send all these notes out now to you as well, so you can have a look at them. And this is also recorded as well, so I'll get this sent out to you as well. And I hope you did find that helpful, and good luck for tomorrow. Not confident for transpiration and translocation. That's a topic that keeps taking. Transpiration, if I just come on here, it's the first few points I mentioned right in the beginning. I'm sure I had it somewhere here. Yep, so factors affecting transpiration. It's these ones here. So water essentially leaving the plant is what transpiration is, the leaf in particular, because that's where photosynthesis happens. And in terms of translocate, are you talking about xylem and phloem? So the xylem takes water just up the cell, just in one direction. The phloem's taking the food and nutrients in both directions. I think that's what you're referring to when you're saying translocation. Yeah. So xylem and phloem, make sure you know which direction they're going in. Xylem's just going up, phloem in both directions. Xylem's just for the water, and phloem's for the food and the nutrients. If there's one I would say to be really particular on and go over as well, the stomata and guard cells for sure.